Hey there, science fans! Today, we're diving into the fascinating world of DNA transcription. It's a process that happens every second inside your body, and it's crucial for life as we know it. Think of your DNA as a giant instruction manual. This manual holds the blueprints for building and running your entire body. However, the instructions are written in a language that your cells can't directly understand. That's where transcription comes in. Transcription is like making a copy of a page from that instruction manual. But instead of just copying the words, we're transcribing them into a different format, one that your cells can read and use. This process is essential because it allows your cells to access the genetic information stored in your DNA and put it to work. Before we dive deeper into transcription, let's take a moment to appreciate the star of the show, DNA. This incredible molecule is the blueprint for all living organisms, carrying the genetic instructions that make you, you. DNA stands for deoxyribonucleic acid, and it's a long, twisted ladder-like molecule found in the nucleus of almost every cell in your body. Imagine it as a tightly coiled thread that holds the secrets of life. The rungs of this ladder are made up of 4 different chemical bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases are like the letters of an alphabet, forming the words and sentences that tell your cells what to do. These bases pair up in a specific way, A with T and G with C, to form the rungs of the DNA ladder. This pairing is crucial because it ensures that the genetic code is copied accurately during cell division. The sequence of these bases along the DNA molecule acts as a code that holds all the instructions for building and running your body. Think of it as a complex recipe book, where each sequence of bases provides a different set of instructions. It's like a language where the order of the letters determines the meaning of the words. In this case, the words are the instructions for making proteins and other essential molecules. In DNA's case, the order of the bases determines the instructions for building proteins, which are the workhorses of your cells. These proteins perform a vast array of functions, from building cellular structures to catalyzing biochemical reactions, making them essential for life. Now let's talk about RNA, or ribonucleic acid. RNA is like DNA's messenger. It's a single-stranded molecule that carries genetic information from DNA to the protein-making factories of the cell, called ribosomes. Think of RNA as a messenger carrying a copy of a recipe from a cookbook, DNA, to a chef, ribosome, who can then use it to bake a delicious cake, protein. There are different types of RNA, but the one we're interested in today is messenger RNA, or mRNA. mRNA is created during transcription, and it carries a copy of the DNA instructions from the nucleus to the ribosomes. Let's break it down. Transcription is a complex process, but we can break it down into 3 main stages: initiation, elongation, and termination. During initiation, the enzyme RNA Polymerase binds to a specific region on the DNA molecule called the promoter. The promoter acts like a start signal for transcription. Elongation is where the real action happens. RNA polymerase moves along the DNA strand, reading the sequence of bases and adding complementary RNA nucleotides to build the mRNA molecule. Finally, during termination, RNA polymerase encounters a stop signal on the DNA, which tells it to release the newly synthesized mRNA molecule. Section 5, calling all builders, RNA polymerase to the rescue. We can't talk about transcription without giving a shout-out to the amazing enzyme that makes it all happen, RNA polymerase. RNA polymerase is like a tiny molecular machine that plays a crucial role in transcription. It's responsible for binding to the DNA, unwinding the double helix, reading the DNA sequence, and adding the complementary RNA nucleotides to build the mRNA molecule. Think of RNA polymerase as a scribe carefully copying down information from a scroll, DNA, onto a piece of parchment, mRNA. Without RNA polymerase, transcription wouldn't be possible, and our cells wouldn't be able to access the genetic information stored in DNA. Section 6, Stage 1, Initiation. Finding the starting line. Imagine you're a runner in a race. Before you can start running, you need to know where the starting line is, right? You can't just start anywhere. You need a clear marker. Transcription works in a similar way. It's a precise process that requires a clear starting point. During initiation, RNA polymerase needs to find the starting point for transcription on the DNA molecule. This is crucial for accurate gene expression. This starting point is marked by a specific sequence of DNA bases called the promoter. The promoter is like a signpost that guides RNA polymerase. The promoter acts like a start signal for RNA polymerase, telling it where to bind to the DNA and begin transcription. Without this signal, the process can't start correctly. It's like a green flag waving at the beginning of a race. This signal ensures that everything starts at the right place and time. Once RNA polymerase binds to the promoter, it separates the two strands of the DNA double helix, creating a transcription bubble. This separation is essential for the next steps. This bubble exposes the DNA bases, allowing RNA polymerase to read the genetic information and begin building the mRNA molecule. The exposed bases are like a code that RNA polymerase deciphers to create a new strand of mRNA. This mRNA molecule will later be used to produce proteins, which are vital for countless cellular functions. So, finding the starting line is just the beginning of a complex and fascinating journey in the cell. Section 7, Stage 2, Elongation, building the message. With RNA polymerase bound to the promoter and the DNA strands separated, it's time for elongation to begin. This is where the magic of transcription really happens. The intricate dance of molecules begins to unfold. During elongation, RNA polymerase moves along the DNA template strand, reading the sequence of DNA bases one by one. This movement is precise and methodical. As it reads each base, it adds a complementary RNA nucleotide to the growing mRNA molecule. This addition is like assembling a puzzle, piece by piece. Remember how the DNA bases pair up in a specific way, A with T and G with C? This pairing is crucial for accurate genetic information transfer. RNA follows a similar rule, except that instead of thymine, T, RNA uses uracil, U. This small change is vital for RNA's unique functions. So, when RNA polymerase encounters an adenine, A, on the DNA template strand, it adds a uracil, U, to the mRNA molecule. This substitution ensures the correct message is built. This process continues, with RNA polymerase adding one RNA nucleotide at a time, building the mRNA molecule like a chain, until it reaches the end of the gene. Each nucleotide added is a step closer to completing the genetic message. Once the mRNA molecule is fully formed, it will carry the genetic instructions needed for protein synthesis, marking the end of the elongation stage and the beginning of a new chapter in the cell's life. Section 8, Stage 3, Termination, time to stop. Just like a runner needs to know when to stop running at the finish line, RNA polymerase needs to know when to stop transcribing the DNA. That's where termination comes in. Transcription termination is triggered by specific DNA sequences called terminator sequences. When RNA polymerase encounters a terminator sequence, it recognizes it as a stop signal. This signal causes RNA polymerase to detach from the DNA template strand and release the newly synthesized mRNA molecule. The mRNA molecule is now free to leave the nucleus and travel to the ribosomes, where it will be translated into a protein. And that's a wrap on transcription. We've successfully created a messenger RNA molecule that carries a copy of the genetic instructions from the DNA to the protein-making machinery of the cell. Section 9. Transcription in action, from genes to you. Transcription might seem like a complicated process, but it's happening right now in every cell in your body. It's the foundation for everything from the color of your eyes to your ability to digest food. For example, the gene that determines your eye color contains the instructions for making a protein called melanin, which gives your eyes their pigment. During transcription, the DNA sequence of the eye color gene is transcribed into an mRNA molecule. This mRNA molecule then travels to the ribosomes, where it's translated into the melanin protein. The amount and type of melanin produced determine the color of your eyes. So you can thank transcription for your beautiful brown, blue, green, or hazel eyes. Section 10. The big picture. Why should we care? Understanding transcription is crucial for understanding how life works at its most fundamental level. It's also essential for medical and scientific research. By studying transcription, scientists can learn more about how genes are regulated, how diseases develop, and how to develop new treatments for genetic disorders. For example, mutations in genes that control transcription can lead to various diseases, including cancer. By understanding how these mutations affect transcription, scientists can develop targeted therapies to correct these errors and potentially cure diseases. Section 11. Keep exploring. The adventure continues. That's it for our journey into the world of DNA transcription. We've learned what transcription is, why it's important, and how it works, all while having a little fun along the way. But the adventure doesn't end here. There's still so much more to discover about the fascinating world of genetics. Keep asking questions, keep exploring, and never stop learning.